All right, Exercise class. Well, we're going to go ahead and dive back into the acute responses to exercise. And so acute response to the cardiovascular and respiratory systems. We're going to just talk a little bit about the AVO2 difference here to start out. And that AVO2 difference, you'll get into that a lot more in the TBL stuff that you study in order to prepare for the IRAT and TRAT on Wednesday. So you'll dive into that more, but just wanted to give you a little brief intro of what it is and kind of how to visualize it. And then we'll get into the respiratory system and its response to acute exercise a little bit after that. So let's talk about the AVO2 difference first. So this AVO2 difference, when we see the A, that A actually stands for artery, okay? And that V is not the same as VO2, like VO2 max. V and VO2 max is for volume. This small v is for vein, okay? And so when we're looking at AVO2 difference, we're looking at oxygen content in arteries versus the veins, okay? And so the idea would be like this. Let's picture a big artery coming down, okay? All this the artery side. Okay, that's going to split off. We'll just go right to the capillary bed, even though we would kind of get smaller and smaller and smaller in our, go to our arterioles, smaller arteries, things like that. Okay, so we got capillary bed, and then we'll have a big vein coming on this side. Okay, so this is capillaries. Okay, so the blood's going to be coming this way, right? It's going to get into the capillary beds, and then oxygen is going to get pulled out. All right, oxygen gets pulled out, okay? And then blood continues on this way back to the heart. That's my little heart. I know it's not a very good heart, but there's my little heart. Okay, so blood's coming this way from the heart, going through the capillaries, getting oxygen extracted, getting back to the heart with blood that has oxygen extracted from it. Okay, so let's just use this for example. We can say that maybe about 20 mils of oxygen per deciliter of blood go in on the artery side. Okay, let's say at rest, as those capillaries as the blood gets into the capillaries and the cells in that area are demanding some oxygen, okay, they're pulling oxygen out. We get about -5 mils of O2. So that's the oxygen that comes out, might be about 5 mils. So what we end up with over here is about 15 mils of O2 per deciliter. Okay, don't worry too much about the deciliter part, it's not a big deal. But it's just this idea that we got a certain amount of oxygen going in, a certain amount gets extracted, and then a certain amount comes back, right? And so if we look at AVO2 difference here, we would say 20 minus 15, right? This is the A part, and this is the V part. So AVO2 difference would be 5 mils of oxygen, okay? And what that represents is oxygen demand, right? Which is really, it's measuring oxygen extraction. Okay, so it's measure extraction, but kind of what it represents is demand. Right, so the more oxygen those cells need, then the more that's going to get pulled out, the higher the demand is. And the way we would see that is by the more was extracted, and that AVO2 difference will be bigger. Okay, so when we were exercising intensely, we might see something like this. We're still going to see the 20 going in, but instead of just the 5, we might see 15 mLs of O2 getting pulled out. And then on this side, we wouldn't have 15 left, we would have 5 mLs left. So that AVO2 difference, AV, we'd have 20 still on the artery side, but only 5 on the venous side. That's going to give us 15 mils of O2. So that might be something we see during exercise. This might be something we see during rest. Okay, so that's the AVO2 difference. And we'll talk a lot more about that during the TBL day, and you'll study it more before you come to class on that day. All right, well, let's jump over to talk about the respiratory system a little bit. And there's going to be some more calculations here, some calculations of partial pressure. Okay, so when we think about partial pressure, first of all, what we need to think about is total pressure. Okay, so when we think of atmospheric pressure, okay, that is going to be represented by pretty much the weight of the air that's on top of our heads. Right, so air on top of us. Right, so when we're down at sea level, the atmospheric pressure is much higher than it is when we go up to the mountains, right? Because there's less space between our heads and the top of the atmosphere, okay? And so there's less weight of air on top of us, okay? So that would be our total pressure, right? But when we talk about partial pressure, what we want to do is we want to look at that total pressure, and by knowing the percentage of each gas that makes up that air, then we can know how much each gas contributes to that total pressure. Okay, so how much each gas contributes to total pressure. Okay, so it's easy just to go through a calculation. So we'll use this example. The atmospheric pressure at sea level is typically right around 760 millimeters of mercury. So that's total amount of weight of air on top of our heads. Okay, the three main gases that we'll talk about, which are the three main gases in the air, are nitrogen, oxygen, and CO2. Okay, so nitrogen makes up the vast majority of the air. Right, it's totally inert. It goes into our lungs, comes back out. Nothing really gets into our body and our bloodstream, nothing like that. There's no diffusion of it. Just comes in, goes back out. Okay, so it's almost 80% with 79. Okay, oxygen is right around 21%, or right at 20.9. Okay, I'm going to tell you right now, you need to remember this number. Remember that percentage of oxygen. You'll need to know that for the exam for things moving forward. Okay, and then carbon dioxide is a small percent, only 0.03. Okay, so how do we get partial pressures? It's a pretty simple process, really. We take our total pressure, so we take our 760, and we multiply it by the decimal form of those fractions there, of those percentages, right? So if we want to get nitrogen, multiply by 0.7904. We want to get the partial pressure of oxygen, multiply by 0.2093. Partial pressure of CO2, and move that decimal place 2 to the left, right? Okay, and that is going to be 0.0003. Okay, and there's the answers to those, right? So about 600 millimeters of mercury, about 159, and about 0.2. So those are the partial pressure of each gas in the air. Okay, and so why do we care about that? Well, we care about that because of this situation here that we have inside of our bodies called the oxygen cascade. Okay, so the idea here, the overall idea of the oxygen cascade is that we have, in order to get oxygen all the way from the atmosphere down to the mitochondria where we can use it in oxidative phosphorylation, every step in that kind of chain of steps has to be a drop down in partial pressure in order for oxygen to move. Okay, and this is just simple diffusion. In diffusion, we know that a chemical or a molecule or a substance of some sort will diffuse from an area of higher concentration to lower concentration, right? And the way that we measure concentration of a gas typically is partial pressure, okay? So when we breathe that air in, we might say that we've got around 150 millimeters of mercury of oxygen, okay? Here we said it was 159, okay? So it just means our partial pressure in this example is a little bit, our atmospheric pressure is a little bit lower than the 760, okay? But that percentage of oxygen does not change, okay? So we go from 150 in the inspired air. By the time we get it down to the alveoli, it's only 104. That's because it mixes with our residual volume, right? Remember that residual volume, that's the air we can't get out. That air isn't fully saturated with oxygen, so the air that comes in has to mix with that, and that dilutes the air of the oxygen in the air a little bit, okay? Now we need to get that oxygen out of the alveoli into our blood, so to load it up onto our hemoglobin in our arterial blood, And so then we drop down to 95. Okay, so there's a little bit of a step there, so that's good. Okay, now that blood goes back to the heart, gets pumped out of the aorta, out of the arteries, down to the capillaries. Okay, now the capillaries got the blood cell. Okay, now the space around those capillaries where the oxygen needs to get to the cells, it's got to first get through that interstitial space, right? And so that's kind of the hardest part. That's the bottleneck in this chain of events, because there's nothing special that carries oxygen through that interstitial space. It just has to float through there, it has to diffuse through. So that's usually where we have the biggest drop, right? We go from 90 to 40. That big drop helps us push oxygen through that space that is hard to get it through. Okay, and then inside of the cells, we expect to see about 20 millimeters of mercury. Okay, and so we have a nice drop there, getting from the interstitial space down to the cells. Okay, and then myoglobin is that protein that we find only inside a muscle, that just similar to hemoglobin, but much higher affinity for oxygen. Okay, and so that's going to be a little bit of a drop down there, so we can get it to the myoglobin. And then now to get from the myoglobin down to the mitochondria, we need another drop. And there's a little bit of variation in how much oxygen is in the mitochondria, right? It can be anywhere from 5 to 0.05, almost drop down to 0. So the fact that it can get so low is protective, right? Because no matter what, if that mitochondrial partial pressure of oxygen can almost get down to 0, that means that no matter what, we're going to get oxygen to the mitochondria, right? We're going to be able to push it there because of the fact that there's always going to be a gradient, right? We have to have a drop down from the myoglobin or from the cells to the mitochondria. And so we got that nice push. Part of that's because of the surface area, right? And so surface area of the mitochondrial membrane is 500 times greater than that of hemoglobin, okay? So the more surface area is, the more the oxygen is spread out, right? The more it's spread out, the lower that partial pressure is. Okay, so let's look at a couple of these questions here. How does high altitude affect each step along the way? Okay, well, let's look at this example right here. So this is a picture of Mount Everest, okay, highest mountain in the world, all right? Still got 21% oxygen, you notice, okay? That doesn't change. What changes is our total atmospheric pressure. Instead of 760, now we're down to 250, right? So 250 is our total atmospheric pressure. So we got to multiply that by 0.21 to get our partial pressure of oxygen. We've only got 52 millimeters of mercury in the inspired air. Okay, so let's go down the cascade using this as our number now. Okay, so instead of 150, we got 52. Okay, and so since we had about a 1/3 drop between the 150 and the 52, okay, right, if we times 52 by 3, it's about 150. Okay, so since we had a 1/3 drop, we'll use that again as a 1/3 drop, or about 30% of 104. Let's call that 35. Okay, so we still got a drop, all right? But we've got to have this, every location down the chain has to get lower, right? So we go from 95, we got to get that down a little bit in order to get, got to get that down a lot in order to get oxygen to move from a space that has 35 millimeters of mercury down, right? So we're probably looking at around 30 here, okay? And then to get to that interstitial space, this has to be the largest drop. So a third of 40 is probably somewhere around 13, right? Let's call it 14, just to kind of round off there. Okay, so 14. Now we had about now we need to drop again about a third of the 20 there, and a third of the 20 is what, maybe 7, right? We'll call that 7. Okay, and then a third of 8 is about, we'll call it 3, just to, not exactly, but we'll call it 3, right? And then we've got to get that mitochondrial partial pressure very low, right? So now we're hoping it's going to be somewhere around 1, okay? But the problem, so the good part about this is we can still get oxygen down, right? We still are moving oxygen, okay? Each step of the way, we still get it down to the mitochondria. So we can do that at the top of Everest. The problem is the amount of oxygen down here at the mitochondria is going to be less than it would have been if we had more oxygen to begin with, right? Because every step along the way, so every step decreases O2 content. Every single step results in lower oxygen content. So what does that do? That eventually means less O2 in mitochondria. And what does that mean? That means less ability to use that oxidative system. Oxidative system. Okay, so we're more and more reliant on the anaerobic system. Our ability to produce ATP with the oxidative system just drops as we get less and less oxygen inside the mitochondria. Okay, so this other question here, what effect would increasing mitochondrial activity have on the partial pressure in the mitochondria? Okay, so if all of a sudden we start exercising, right, and our mitochondria become very active, we start using up all that oxygen, that's going to drop the partial pressure, right? So partial pressure of O2 will go down, right? So if we go back to the right side, we start back up here with 150, we get all the way down here, we're going from 8 down. Instead of 5, like it was before we were exercising, maybe that drops it down to 2 or 3, okay? So what happens? That means more O2 moves, right? Because the stronger the gradient we have, the more oxygen is going to move, okay? So decreases partial pressure, or sorry, that increase in mitochondrial activity, right? That's going to decrease partial pressure of oxygen, and it's going to result in more oxygen diffusion, okay? And so what does that actually mean? For the amount of blood that comes, amount of oxygen that comes back, if we're going back to our AVO2 difference idea, right? What effect on the partial pressure, what effect does this have on the partial pressure of the venous blood? It's going to be lower, right? So if there was more oxygen diffusion, or if we look at that AVO2 difference, A is going to stay pretty much the same because we're not desaturating our arterial blood, but that V is going to be lower, right? And so if that V is lower, then our AVO2 VO2 difference is higher, all right? So we increase AVO2 difference when we start using up oxygen in the mitochondria because there's a stronger diffusion gradient. Something you got to sit with and work with a little bit, but very, very important concept for understanding this. Okay, this is just kind of looking at that oxygen cascade, same way we had before. The only thing this shows differently is we need a cascade for CO2 as well, but it's got to be the opposite. The highest point of CO2 concentration needs to be out here in the tissues, right? So we can push CO2 onto the blood, right? And so we go from 45 to 40 here, okay? And then that 40 is going to travel back, right? And now we've got, sorry, that's the wrong one. I switched over to the oxygen one. 45 to 45 there. Inside here, it's a little bit higher, right? It's 46, so we go from 46 to 45. And then that 45 is going to bring us back up. 45 here, only 40 in the alveoli, so it's going to allow CO2 to diffuse across. Okay, so there's got to be a gradient for the gases to move, both oxygen and CO2. Okay, moving on to a different concept here, another crucial one to understand for the AVO2 difference and for how our body is able to get more oxygen off of the red blood cells, okay? And so again, when you're thinking about your concept map, we talked about the heart rate side, we talked about the stroke volume side, now we got to talk about the AVO2 difference side, right? And so one thing I just said that's going to help us increase our AVO2 difference when we're exercising is we increase mitochondrial activity, right? Because that's going to increase oxygen diffusion. That's going to result in more oxygen getting off of the blood with a lower venous concentration of oxygen, okay? So that was number one. Another one here that we will see is the way that this curve is affected, okay? So I know some of you have seen this curve before. You've all seen it in anatomy and physiology, but it's always a trickier concept, okay? So using the oxygen cascade figure, find what the saturation percentage is both when the red blood cell is next to an alveoli, okay, I'm reading this right here, and also when the red blood cell is next to the interstitial space of the tissues, okay? So let's go back. Okay, so when the red blood cell is next to the alveoli, okay, we're looking at about somewhere about around 100, right? So 104, okay? When it's next to, what was the other part that I was saying? When the red blood cell is next to that interstitial space, right? Sorry, that was that one. and the interstitial space is 40, right? So we want to look at both the partial pressure at 40 and the partial pressure of 100, okay? So the way you use this graph is we take partial pressure of oxygen down here, that's on the x-axis. We don't care so much about this, to be honest. We're more interested in this side, which is oxygen saturation on this y-axis. Okay, so this is really plotting the relationship between when you have a red blood cell and you put it in solution with a certain amount of oxygen around it, how much oxygen ends up on the red blood cell, how much actually loads up into the hemoglobin. That's what this graph tells us. Okay, so if we're in a situation where oxygen is about 104, like it was right there, okay, that's the situation of the in the alveoli and in those capillaries in the lungs that are going to take in the blood and get it filled up. Okay, so we're going to take this 104, we're going to bring it up, we're going to see where it intersects with this line, and then we're going to take that over, and that's going to give us our saturation. That's about 99% saturated, okay? So you've all had this test on you before. You get that little red thing that clips onto your finger, little red light, right? That's an O2 sat monitor. That's using light to measure how much, what percentage of your red blood cells are saturated with oxygen, okay? So if the partial pressure in your lungs is around 100, which it should be, or higher, then you're going to be 99% saturated, okay? But then here's the interstitial space one. It was 40, right? So if we take that 40, we go up, see where that intersects the line, about there. Okay, we're going to take that over. Obviously, my lines aren't perfectly straight, but we're probably looking at something like 75% saturated. Okay, so between filling up our red blood cells at the lungs, and then by the time they get down to the capillaries, right there at the capillaries, they're going to drop off about 25% of the blood, or 75% of the oxygen, right? So going from 99 to 75, 24% of the oxygen gets dropped off under normal circumstances, okay? And that's how we use this graph, okay? One thing that's really advantageous for us, two things, is the fact that we have this huge plateau up here, right? It's so flat. So that means even if our partial pressure in our lungs drops drastically because we're not breathing very well or at altitude, right? Even if we're down somewhere around 60, that's still going to give us a saturation of around 95, 96, right? That goes all the way from 160 down to about 60. We have full saturation still, okay? And that's a very advantageous thing for us because we can survive under different varying levels of oxygen in the air, right? But then the other part that's advantageous is all of a sudden, right about here, it drops off super steep, okay? And so that means that when we go from 40, okay, or even if we go from 60 down to 40, there's a big difference in how much saturation. But let's say that interstitial space content goes from 40 to just 35, which would probably take us like right here instead. That results in a huge difference, right? That goes from 75 to 60%, meaning that just by a 5 millimeter decrease in oxygen level in the interstitial space, we dropped off 15% more oxygen, right? So that steep part is very beneficial because anytime we're using more oxygen, we're going to decrease that amount of oxygen in the mitochondria, which we backtrack, that decreases the amount of oxygen in the interstitial space. And that is going to result in a lot more oxygen being dropped off. So that's good, right? When we're using a lot, we want to drop off a lot more, okay? So that's an important piece. The other important piece is we can move this thing, right? This curve shifts on us, right? So how do we shift this curve? This curve shifts by 3 different mechanisms, okay? It shifts by the influence of hydrogen ions, or pH, okay? So a high hydrogen ion concentration or low pH results in a rightward shift, okay? So that blue line there, so rightward shift. Opposite, high pH or low hydrogen ion concentration results in a leftward shift, okay? I'll just show you the other ones. Same thing with CO2, high CO2, low CO2. High CO2 is rightward, low CO2 is leftward. And then lastly, temperature. High temperature here versus low temperature. Rightward shift for high temperature, leftward shift for low temperature. Okay, and what does that mean? What does a shift mean? Okay, so and the question is there, what effect would a rightward shift have on AVO2 difference? Okay, we're going to get to that one in a second. But first of all, what does a rightward shift mean? Well, let's just figure it out with the graph, and you guys can do this with me. Okay, so we see 40 here. That's our interstitial space, partial pressure. We took that up to here before to the black line, went over. That gave us about 75% saturation. Okay, but let's say we're producing a lot of hydrogen ion because we're exercising a lot. Okay, so that pH is going to drop, maybe something like that, right? High concentration of hydrogen ions. That drops our pH, so we get a rightward shift. Take that line up. Now we intersect with the blue line instead of the black line. Take that over. 60% now, right? We dropped off 15% more, so increase in 15% oxygen drop off. What does it say? It says better unloading, right? So that means more oxygen is dropped off. So if we drop off more oxygen, when we go through the capillaries, that means that we're going to end up with a lower amount of oxygen on the venous side when we look at that AVO2 difference, okay? So AVO2 difference, right? This, again, the arterial side is going to stay the same. The venous side is going to be lower, which is going to lead to an increase in AVO2 difference. Okay, so that rightward shift would lead to an increase in AVO2 difference. It's going to be just the opposite on the other side. Okay, so we get a leftward shift. If we go this way, so instead of being at 75, like we were here, we get that rightward shift, and now we're at more like 82. Right, so we have less drop off, right? Inhibited unloading. Okay, and so that leftward shift would decrease AVO2 difference. Okay, and so that's a key piece. So that's something else that happens during exercise that helps us change our AVO2 difference, right? We have an increase in hydrogen ion concentration. We definitely have more CO2, right? Because that's a byproduct of metabolism, and we heat up too, right? So all three of these things are direct byproducts of metabolism, okay? And so when we have these direct byproducts of metabolism, so we're using up more oxygen, right? Using up more oxygen, which produces heat, produces CO2, produces hydrogen ions. All three of those things lead to us dropping off more oxygen, which makes perfect sense, right? And that's the wonderful thing about physiology. It makes sense, right? If we're exercising more, we're using oxygen, producing CO2, producing heat, producing hydrogen ions. So all those things result in us dropping off more oxygen so we can use it, okay? I'll give you a little hint. These same things are also vasodilators, right? These things open up capillary beds so we can get more oxygen into the we can get more oxygen into the mitochondria, into the cells, so we can use it as well. And we get more oxygen into the capillaries, which means more diffusion, okay? So far for that AVO2 difference, we've got increased vasodilation or increased open capillary beds. That would lead to a greater AVO2 difference during exercise. We've got higher temperature, higher hydrogen ion concentration, higher CO2 concentration. Those things lead to a rightward shift of this curve, which means more oxygen being dropped off. And then we have higher mitochondrial activity. Okay, all right, guys. Study hard for the IRAT and TRAT on Wednesday, and we'll see you then.